classes. So we'll continue. Today we will learn something from nitrogen metabolism, that is nitrogen nutrition. Nitrogen nutrition, they all the organisms for their important metabolites. Okay, they need nitrogen to synthesize different important components, important biomolecules like protein, nucleic acid, chlorophyll, and some other hormones, etc. So this nitrogen, there are plenty of nitrogen in the air, but this is the nitrogen, the plants particularly, they cannot absorb and utilize. So this free nitrogen is not useful for the plant. We animals, we take nitrogen nutrition from by eating plants and other animals. But the plants, they get it from the nature, not from the air. So it is only a few plants, few organisms, they can directly utilize the atmospheric nitrogen. The other plants, they get this nitrogen when they are present in the form of certain compounds of nitrogen, like the ammonia, nitrate, nitrite. And ammonia is the first stable compound which is metabolized by the living organism, particularly the plant. So this free nitrogen of the air has to convert it into ammonia or nitrate, then the plant can absorb it. This nitrogen fixation, fixation of nitrogen, this is the conversion of, this is actually the conversion of molecular nitrogen, nitrogen into ammonia. So it is converted, and this conversion, that molecular nitrogen into ammonia or some other compound like nitrate and nitrite, nitrate and nitrite. This requires a lot of energy, and some organisms can convert it. In nature, several ways the nitrogen is fixed. Okay, it is non-biological and biological processes there. So we will see those things, and we will practice some questions related on this nitrogen metabolism. Here is one question. The process of conversion of soil nitrate into free nitrogen is called and is carried out by bacteria, options are nitrosomonas. Number 2, denitrification. And nitrobacter. Number 3, option denitrification. Thiobacillus. And number 4 is nitrogen fixation. And this rhizobium. So here, this is the question. These questions are very easy thing, but here require only your memory. So you have to remember that which bacteria or which organism carry out a particular process. Okay, so here the options A, you have to see also the pair, that A is in option 1. This A is the nitrification, B is the nitrosomonas. Nitrification is conversion of soil ammonia into nitrate and nitrite. This is conversion of ammonia into nitrate and nitrite. This is called nitrification. Here the process is the reverse way. That is, not that ammonia is converted into nitrate. Here nitrate and nitrite, they are converted into nitrogen. Okay, so this process is denitrification. The process is denitrification. The conversion of nitrate into nitrogen. It is reduced into nitrogen, and this is denitrification. And those bacteria which carried out this process, that we in general call the denitrifying bacteria. This is denitrifying bacteria. And this denitrifying bacteria are, actually it is the thiobacillus. This denitrifying bacteria are thiobacillus and pseudomonas. Okay, these two are particularly the denitrification they carry out. So these are, we can say, it is harmful that they reduce the soil fertility. Actually, when there is nitrate or nitrite in the soil, the plant can absorb that nitrate and nitrite. Then this nitrate and nitrite within the plant is converted into ammonia, and from the ammonia, the amino acids are synthesized, and then amino acid is utilized for the synthesis of protein. So plant gets from the soil, plant gets the nitrogen nutrition in the form of nitrate. And this bacteria, denitrifying bacteria, it converts the nitrate, this nitrate into free nitrogen. And when it is free nitrogen, this is released in the atmosphere. And again, that free nitrogen is not useful for the plant. So this here our answer is that this is denitrification. So this conversion of soil nitrate into free nitrogen is called denitrification and is carried out by bacteria thiobacillus. So this is not the process of nitrogen fixation. Nitrogen fixation is conversion of free nitrogen into compound of nitrogen, which can be utilized by the plant. So it is the ammonia and the nitrate and nitrite. This is here. This one is the nitrification. Nitrification is carried out by nitrosomonas. Nitrification is conversion of ammonia into nitrate and nitrite. So this nitrification is carried out by nitrosomonas as well as nitrobacter. Okay, nitrobacter convert this nitrite into nitrate, and this nitrosomonas this convert the ammonia into nitrite. So this process is nitrification. This in nature that the organic matter of the plant and matter of plants and animals. Animals, that is after the death, the dead organic material of the plants and animals, they are into converted into ammonia. Okay, and this process is known as ammonification. So in the nitrogen metabolism, ammonification is one of the methods. This and once this ammonia is formed, this soil ammonia is converted into nitrate and nitrite. It is absorbed by the plant. Nitrate and nitrite is also formed when there is in the that electric spark during this rainy season, thunder, and this at a high temperature, this molecular nitrogen, they combine with the atmospheric oxygen. Okay, at the time of lightning, when there is during the rainy season, there is thundering, lightning, and in that process there is very high temperature is produced, and at that high temperature, the nitrogen combined with the oxygen, and they form the oxides of nitrogen. This oxides of nitrogen get dissolved in water and form the acid, nitrous acid and nitric acid. And this nitric acid and nitrous acid come to the ground in the soil, and then it is gradually converted into the nitrate and nitrite, and then nitrate and nitrite is absorbed by the plant. So in nature also, that non-biologically, there is a conversion of nitrogen into nitrate and nitrite, and there are some organisms which also convert this molecular nitrogen into nitrate, nitrite, and ammonia. Okay, this is this nitrogen fixation. Nitrogen fixation is a process that is solely carried out by prokaryotic organism only. It is only carried out by certain prokaryotic organisms. No eukaryotic organism can fix the nitrogen. Okay? So within the prokaryotic organisms also, not all prokaryotic organisms, it is certain bacteria and some blue-green algae, that is, and some cyanobacteria. The blue-green algae. In this cyanobacteria, very important one is Noxa and Anabena. They can fix the nitrogen as a free living as well as symbiont, that is living in the body of other organisms. There also it can fix the nitrogen or it can fix the nitrogen in free living condition. In bacteria also, some bacteria, they can fix nitrogen as a symbiont as well as some bacteria as a free living. The most common symbiotic nitrogen fixing bacteria is the rhizobium, which forms the root nodules in all leguminous plants. Rhizobium bacteria, this is a nitrogen fixer as symbiont which form root nodules in leguminous plants. That is, in case of pulses, that gram, pea, beans, clover, okay, alfalfa, all these different leguminous plants, they form the root nodule. So this rhizobium bacteria stay in the root nodules of this leguminous plant and fix nitrogen. There is another, that filamentous bacteria is there, which is known as Frankia. Okay, this Frankia form root nodules in non-leguminous plants. Plants like Alna casuarina, etc. So this one forms, these are the tree species, but the rhizobium, which they are infecting the herbs. Most of this leguminous plants are mostly their herbs. There are some trees are also there, but this Frankia, they are producing root nodules in the tree species, and it is non-leguminous. Next question is rhodospirillum. Rhodospirillum is here A, nitrogen fixing, B, anaerobic, C, free living, D, photosynthetic, and E is aerobic. Now the answer options are: number 1 is A, B, and C. Number 2, A, B, and E. Number 3, B, C, and D. And number 4 is A, B, C, and D. Which of the options, what are the characters that should form this rhodospirillum? A is nitrogen fixing, anaerobic, free living, photosynthetic, aerobic. So option 1 says A, B, and C. That is nitrogen fixing, anaerobic, free living. This is, of course, this rhodospirillum is true that it is the nitrogen fixing bacteria, and it is anaerobic as well as it is free living. It lives independently, but at the same time, the rhodospirillum is a green bacteria and it carries out photosynthesis. So this one, that all these four characters, that nitrogen fixing, anaerobic, free living, photosynthetic, this is correct. So our option of answer is number 4. It is not aerobic. Aerobic and anaerobic, that is the bacteria which for its respiration require oxygen or not. Those bacteria which are dependent on oxygen, they are the aerobic bacteria, and which is independent of oxygen, they are anaerobic. Here match column 1 and column 2 and select the correct option. One, column 2. A denitrifying bacteria B aerobic bacteria C anaerobic bacteria and D is cyanobacteria. Beside 1, 2, 3, 4. One is Thiobacillus, number 2 Azobacter, number 3 Rhodospirillum, and number 4 is Nostoc. Now the answer option, number 1, number 2, number 3, and number 4. A 3, B 2, C 1, D 4. A, B, C. Oh, sorry. Okay, so from here we have to select the correct match. This year already we have, in the previous question, we have learned that this thiobacillus is denitrifying bacteria. This converts that nitrate into free nitrogen. This is, so this one is matching with this. Azobacter is an aerobic. Rhodospirillum, we have done already one question where that we have learned that rhodospirillum is anaerobic bacteria. This one is also correct. And this is cyanobacteria. Nostoc, already I have told that the nitrogen fixer, symbiotic as well as free living, Nostoc and Avena, they are the cyanobacteria. So it is already that given column, this is already matched. Okay, this thing confuse us sometimes, test our confidence that how can in the question it is given all correct match, but in this question it is there. The column is already, that given column is already matched. So this will test our confidence in our knowledge. So this is here that option number 3, where is the A1, B2, C3 and B4, this is the answer. Okay. Next. Here, which of following statement is incorrect? With respect to the pigment leg hemoglobin, this is a pigment, very, very important pigment in the process of nitrogen fixation. In the process of nitrogen fixation. And this pigment is found in all the plant tissues where these bacteria, they are living and synthesizing this ammonia from nitrogen, that is converting nitrogen into ammonia. So nitrogen fixation is there. So this statements are: number 1, it is an oxygen scavenger. Number two, it is pinkish in color. Number three, it catalyzes the function of nitrogenase under anaerobic conditions. It combines with oxygen and protects nitrogenase. So here, in relation to regarding the leg hemoglobin pigment, which statement is not correct? This is a pigment which is found in the root nodules in the host cell of root nodule of nitrogen fixing those the host cells of root nodule. That is the root nodule cells where the bacteria rhizobium fix the nitrogen. Actually, the process of nitrogen fixation, the biological nitrogen fixation particularly, biological nitrogen fixation occurred in absence of oxygen, in the environment where oxygen should not be there. So in anaerobic condition, okay, not in the aerobic condition, because the enzyme that nitrogenase is the enzyme which is important for the nitrogen fixation. Okay, this nitrogenase enzyme is very sensitive to oxygen. So if oxygen is there, then the nitrogen will get oxidized and it will get spoiled. Okay, because this nitrogen fixation is a reduction process, is it? And this is oxygen. If oxygen is there, then maybe reduction will not take place. It oxidation will take place because nitrogen is converted into ammonia. So here the hydrogen is added. So this process is a reduction process, nitrogen fixation. So, and this takes place in absence of oxygen. So for the nitrogen fixation, this bacteria, this enzyme require an environment which is free from oxygen. And this environment is provided by the pigment leg hemoglobin. Okay, this is provided by leg hemoglobin. This is the leg is for leguminous, abbreviated. This is the leg. Okay, and hemoglobin, like in our blood also, there is pigment hemoglobin which carry oxygen. This is very much sensitive again. It can catch oxygen. So there is within the root nodule, the tissues where the bacteria live, they have a pigment, and as this pigment is found in the leguminous plant root nodule, so this has been the given name leg hemoglobin or leguminous hemoglobin. This leguminous hemoglobin is like the hemoglobin, normal hemoglobin, that this is also slightly red in color, but not exactly red. This is pink. Leg hemoglobin is pink in color. So this it is a pinkish in color. This one is correct. Okay, and due to this leg hemoglobin, the root nodule, color of the root nodule is slightly pink. Okay, and this leg hemoglobin, they are very sensitive, and this catch the oxygen. So it is oxygen scavenger. It destroys oxygen from this environment, from the tissue. So what it does, it combines with oxygen and protects nitrogen. So it combined with oxygen. So naturally it is a oxygen scavenger, it is called. It is pink in color, and this statement also that it is saving the nitrogenous by combining with oxygen, so make the cell environment oxygen free. And say this one, that this statement, it catalyzes the function of nitrogen fixation. Nitrogen fixation in anaerobic condition. This one is not correct. This is statement regarding the leguminous hemoglobin. So this is which is incorrect. So the incorrect statement, the wrong one is right here. Okay? So it's catalyzed. This is not true. This is not correct. This is not a catalyst of the process of nitrogen fixation, or for the, it helps the nitrogenous in other way, but not catalyzing the process of combustion. Okay. Here the amino acid which plays a central role in nitrogen fixation or in nitrogen metabolism is uh, option number 1, glutamic acid, 2, alpha keto glutaric acid, 3, aspartic acid, 4, double aminated with keto acid. The amino acid which played a central role. Actually, this in the process of nitrogen fixation, that nitrogen is converted into ammonia. This ammonia is changed into ammonium ion. It is called protonated. It is ammonium ion. And then this ammonium ion supplies amino group, that is NH2, this amino group. The amino acids, all the amino acids have at least one amino group and one carboxylic group. Okay, one carboxylic group and one amino group. So this amino group is supplied for the synthesis of amino acid. This amino group is supplied by this ammonium or ammonia. Okay. To some carboxylic acid. To some carboxylic acid, which is already having that carboxyl group. The amino group is not there, so to that amino group is added and an amino acid is formed from the organic acid. This is when that is the first amino acid which is formed in this process is the glutamic acid. Is the glutamic acid, and it is produced from alpha ketogluteric acid. That is alpha ketogluteric acid to this is added ammonium and NADPH ammonium plus NADPH, okay, and it forms the glutamic acid. From this glutamic acid, this is an amino acid, and from this glutamic acid, other amino acids are produced. So, from the organic acid, amino acid, glutamic acid is synthesized, the amino acid glutamic acid is synthesized, and then glutamic acid can transfer or supply the amino group for the synthesis of other amino acids. And that is why the glutamic acid is the one which plays a central role in the nitrogen metabolism. Okay, so glutamic acid is the first amino acid synthesized, and this is playing a central role also. This aspartic acid also is another amino acid. Okay, this double aminated, these are actually the amides, and this is produced by the process that is called the reductive amination. And that is transamination. Okay, by this process, the amino acids are synthesized. Two process. In the transamination, the amino group from the glutamic acid is transferred to other carboxylic acid, and then it is synthesized other amino acids. By reductive amination, glutamic acid is synthesized. So first step is the reductive amination, and second step for the synthesis of other amination is transamination process. Okay, thank you for today's class. Bye.